All right, sounds good. Thank you, Liz. I'm happy to be here. Good afternoon, everyone, or morning if you're joining us from the West Coast. And I have literally been a VMO for 15 days now, but I've really enjoyed being a part of these fab schools for going on 2 years, and I really like poultry. So that's where this presentation comes to me. Avian influenza, Newcastle disease, very likely you'll be dealing with these diseases. And as our paddle director, Dr. Holland, pointed out in her presentation, you don't send these samples to FADL, you actually send them to the Diagnostic Virology Lab in Ames, Iowa. Nevertheless, the important diseases you'll likely be dealing with, and we actually get to see Newcastle disease in the in-person training. So I'd certainly like to acknowledge the colleagues at NBSL whose slides I adapted this presentation from. And we'll get started. So this presentation to me is all about telling you how to talk the talk. And then in the in-person training, we'll work on the walking the walk with the sample collection. But some of you may be influenza experts, but I'm going to just give a brief overview of avian influenza to start. And after I go over the history of virology, epidemiology, clinical findings, then we'll wrap it up with diagnostics and control and switch to Newcastle disease, two very different diseases with similar clinical presentations, and we'll discuss that. Just to start with some history, does anyone know what this picture depicts? You can put it in the chat or you can yell it out, or if you don't know, we can talk about it. But it's hard not to start an influenza discussion with, yes, a picture of the 1918 Spanish flu pandemic. So a lot of you probably know Spanish flu is kind of a misnomer. It didn't start in Spain. It had to do with lack of censorship during World War I. So Spain was actually reporting their cases. Believe it or not, it started right down the road from where I'm at right now at the National Bio and Agri-Defense Facility at Camp Funston. This is in Fort Riley, Kansas, where the first cases were documented. Something you may not know is that when they sequenced this flu strain years later, they found out that it was actually of avian origin, or it had similar genes to avian-like flus. And of course, that's what the topic of discussion today is. So highly pathogenic avian influenza is a devastating disease. You can imagine when the clinical sign, most common clinical sign is sudden death, that this is just a really hard disease to deal with. And being a producer, seeing, first of all, having a non-negative test at your farm and then having to deal with the depopulation, it's really devastating. So this is just an example of morbidity and mortality in a commercial turkey flock with highly pathogenic avian influenza. Whoa. So avian influenza, it's an orthomyxovirus. It's a type A influenza virus. Humans get type A, B, and C. There's also type D that swine and cattle get, but avian influenza is a type A influenza, so sometimes you'll see it abbreviated as type influenza A virus. It's an enveloped RNA virus, single-stranded, negative sense with 8 segments, and you're likely familiar with the major surface proteins, hemagglutinin and neuraminidase, and different subtypes of these. There could be 144 possible combinations of these surface proteins, so that's one way we type it, which I'll go into. So this is all microbiology jargon, right? And we're all veterinarians. So what I like to do is try to translate this from science jargon to what we need to know about this virus to complete an FAD investigation, respond to outbreaks, and communicate with certain stakeholders. So I kind of simplify it. From being a type A influenza virus, we know that all flu pandemics have been caused by type A influenza, including the 1918 flu pandemic. That was a type A influenza. I always like to think of envelope viruses as unstable. It's kind of a paradoxical thing, but we know these types of viruses are more unstable at high temperature, which is why we can compost and we have those temperature targets, or when exposed to chemical disinfectants. The R in RNA stands for rapidly evolving in my mind. So they have a high mutation rate and error-prone polymerase during replication, and genome reassortment is how new subtypes and even pandemic influenza emerges. And that's where these different subtypes come into play and how certain outbreak strains are referred to, such as H1N1 in 2009. So it's important to know how to name these. It starts with the virus type. So again, we're dealing with influenza A, the place it was isolated, the strain number, the year of isolation, But the most familiar thing to everybody is going to be this subtype. And here it's H3N2. If we look at all of the avian influenza pandemics, or sorry, not pandemics, but outbreaks that have occurred since we started typing these things, that a really interesting pattern emerges. And that's that they're all H5 or H7 subtypes. So, and that has to do with the way that the World Organization for Animal Health defines these influenza viruses. So the WOA definition for this is for reportable, highly pathogenic avian influenzas, any influenza A virus that has been determined as highly pathogenic. Now, the gold standard, but kind of old school way of doing this was an intravenous pathogenicity index. You take a few chickens that are SPF chickens, you inject them with live virus, and you just look and see, and if a certain percentage of them die, you know it's a highly virulent strain, therefore it's HPAI. Nowadays, we do diagnostics and a lot of sequencing to determine this. So we can actually sequence this at NVSL AIMS and determine if it has these multiple basic amino acids in the HA molecule. And also, it's important to point out that even low pathogenic avian influenza viruses of the H5 or H7 subtype I just described are also reportable, just due to all avian influenza outbreaks being those subtypes and the potential for those to turn into outbreak strains. We'll get more into the details of this when we talk about Newcastle, but this is this. This is a very busy infographic depicting the diverse influenza host range. And the highlights here are this native or reservoir host of influenza viruses, which is waterfowl. And then most mammalian and avian species have certain seasonal subtypes that circulate amongst them. Of course, sharing is caring, and reassortment can occur between mammals and birds as depicted here. And this can result in novel strains such as the 2009 H1N1 flu pandemic. Now for here, I think the proverbial elephant in the room is actually a Holstein cow. So this is a situation that's been developing since March. There's been at least one confirmed human case, and we're really learning more and more about this each day. We can absolutely talk about what is known at the end of this presentation at this time, but I really want to focus the training on this as to focus on HPAI and commercial poultry. But I can tell you that poultry producers are really concerned about HPAI and dairy cattle, mainly because it's just one more exposure source for them. So now not only do they have to worry about other infected farms, but they have to worry about dairy farms that may be in proximity to them. So we could talk about that more later, but one thing I want to highlight is how these arrows between different species emerge. And that's through genetic drift and shift. So antigenic drift pertains to mutations in the H gene, which causes minor antigenic changes in the H protein. And this is an ongoing process. It's constantly happening, and it's really a problem for protective and vaccine-induced immunity. On the other hand, antigenic shift is a complete replacement of the H or N gene. And this is how we get a reassorted virus and the insertion of extraneous RNA, and that can result in a virulence shift and emergence of novel or pandemic strains. When I first learned this, I was always getting them mixed up. So I came up with a way to remember it. I like cars. These are both kind of car terms. So I like to think of antigenic drift as kind of like you're drifting a car and your tire tread changes. It gets left on the road, and those are like the little point mutations that happen. The antigenic shift, however, is when you're actually shifting from park to drive, and that could be the shift of one strain to a completely different strain and the emergence of a novel or pandemic strain. So if that helps you remember it, great. If it doesn't, I'm sorry. That's the best I could come up with. So just some basic epidemiology. The reservoir is wild aquatic birds. The incubation for avian influenza can be hours to 3 days. And this is highly dependent on the host species, the viral strain and dose, how old the host is and their immune status, and the presence of other infections, and the environmental conditions as well. The shedding is highly variable. Again, it's strain and condition dependent, but avian influenza is viable for longer periods in cool and moist conditions. And as for transmission, there's the typical routes, fecal oral, but also aerosol transmission is really important for this virus. This is just to review the natural reservoirs are wild aquatic birds. The majority of the influenza subtypes are found in 2 orders, and that's here on the left, mallards, brants, and geese, but also on the right, these are our gulls and shorebirds, such as ruddy turnstones and terns, if you're a birder. I like to think of these as waterfowl versus water adjacent birds. But the important thing to remember is that, these are reservoir hosts and they have limited clinical disease, even if they're infected with a highly pathogenic avian influenza strain. And they fly, so they can spread it pretty much anywhere. And that's another discussion on how the flyways impact the dissemination of this disease. So this fun graphic is just showing the many things you really have to think about when you're dealing with an infected premises. And these things in red, such as infected birds, people, vehicles, and equipment, we can manage, but it's not always straightforward. Infected birds, for instance, they can be depopulated and compost in the house, so you're really limiting where those things go that could spread virus. You can also limit the movement of people on and off farms, but always remember that the vehicles and equipment coming off and on poultry farms can be unexpected. So you have to think about utilities like people who are servicing your propane, power, and things like that. These yellow arrows are difficult to manage. So that includes the wind. Of course, viral particles can be carried in the wind and be spread. Wild birds, which we just discussed as a reservoir host, as well as mechanical vectors such as insects and other vectors such as rodents. So when I was trained to think of this as like an HPAI positive farm, I was trained to think of this like a spider web. It's really complex, but you really truly have to think about all these aspects and the interconnectedness of them when you're dealing with an HPAI case. So we further classify avian influenza into low pathogenicity, which is a localized infection. So this is known as LPAI. It produces subclinical to mild disease. And waterfowl, that disease is limited to the GI tract, and domestic birds, typically the respiratory tract. And again, as a reminder, this is outdated, but the World Organization for Animal Health defines H5 and H7 in poultry as reportable. Sorry, the initialism is outdated, not the information. Those strains are still, those subtypes are still reportable, even if they're LPAI, just because of their, the possibility of them evolving to be a highly pathogenic avian influenza. LPAI has really non-specific clinical signs, and it may not even involve a foreign animal disease investigation. It may be something that's picked up through routine influenza surveillance. But it could be cause decreased egg production, food and water intake, again, sometimes difficult to notice, as well as increased respiratory disease, subclinical, and a slight bump in mortality. Really important to note that these characteristics are not specific to avian influenza. There are many differential diagnoses that can cause these in poultry flocks. And again, you may just figure this out after some serological surveillance down the road. But the reason we're here is for this one, the big one, highly pathogenic avian influenza. This is usually very obvious. You see acute systemic disease with very high mortality. It affects primarily poultry. This includes chickens and turkey and any bird of economic value, and it is a reportable disease. And again, this is no longer constrained to a specific subtype. So what this means is even if an influenza strain that's not H5 or H7 subtype emerges as highly pathogenic, it is reportable. So I want to introduce a fundamental concept that pertains to the pathogenesis and virulence, and we'll expand on this in Newcastle disease because it's a little bit easier to explain. But for right now, I want to introduce this, and you can remember that there are these proteases. They're usually a trypsin or trypsin-like protease in the host cells, not part of the virus. This is in the host, and it's depicted here by this green enzyme. And it's these host cell proteases that cleave this HA protein of the virus. And the cleavage of this protein is essential. It's absolutely necessary for viral replication. So what this means is that for LPAI, cleavage occurs by enzymes only in the respiratory tract or the intestinal tract. And I believe that gets into a question in the chat. Someone can correct me if I'm wrong, but one of the questions was, do we know why it's GI and waterfowl versus respiratory and domestic birds? And that just has to do with waterfowl having more of these proteases in their GI tract, where more viral replication and thus shedding happens. HPAI, on the other hand, this cleavage occurs almost everywhere in the host body. So that means that these specific enzymes are available in many different host tissues. So to take that out of virology jargon and put it into chicken infection jargon, it just looks like this, a different way of looking at it. But again, in LPAI, these proteases are restricted to the respiratory tract as well as the GI tract, whereas in HPAI, you see these proteases everywhere, which means the virus can replicate everywhere and produce severe systemic disease. And again, it basically pertains to the amount, the sequence that's at this cleavage site and the ability of that enzyme to recognize that cleavage site. So that brings us to the clinical presentation of highly pathogenic avian influenza. It could start with coughing or snicking, which is kind of like the bird version of sneezing, if you're not familiar, and open mouth breathing. So this is a huge warning sign when walking into any poultry house. If you see birds doing this, you probably know you have a problem. If not avian influenza, it's another serious problem. Birds may huddle together, they have ruffled feathers, they look depressed, they're just generally poor doing with drooping wings or head. These are more nonspecific. You'll see discharge from the eyes and nares and redness and swelling of the head. And then this is where we get into kind of the more alarming lesions that you'll see when you actually post a bird or perform a necropsy. And those are these hemorrhages in the proventriculus, which often occur on the papillae. With HPAI, there's commonly like a green watery diarrhea. I believe this bird was alive and had diarrhea and died immediately thereafter, not because it was euthanized, but because it was infected with HPAI. So again, sudden death and high unexpected mortality is kind of a hallmark of this disease. And again, you've seen hemorrhage and necrosis in multiple tissues throughout the body. You can see cyanosis of cones and wattles, and as well as subcutaneous hemorrhage. And here on the right is tracheal hemorrhage. That brings up an important differential diagnosis for HPAI as well as Newcastle disease that I'll cover in the end, but there's another disease in poultry that you're probably thinking of that can cause this lesion as well. And then again, we're starting to pick up on a theme here, but this is just a systemic disease that causes hemorrhage in multiple tissues, everywhere from muscle to skin. And then if you see in this pericardial fat, there's multiple pinpoint hemorrhages there. So we'll hammer this home in the Newcastle portion of this talk as well. So for HPAI diagnostics and control, diagnostic testing, this is something we'll really get into in in-person training when I see you at Plum Island, but you're going to be submitting tracheal, oropharyngeal, or cloacal swabs in BHI. And cloacal swabs are something they prefer to submit in waterfowl, again, just because they shed so much virus from their GI tract. And oftentimes this BHI will contain antibiotics to cut down on bacterial growth so they can look for the virus. These samples are going to the Diagnostic Virology Lab at NVSL in Ames, Iowa, and have kind of a different sample submission process that we'll talk about in the future. Diagnostics for this disease are going to be real-time reverse transcriptase PCR. And what they're doing is they're looking for the influenza-specific matrix gene, and then they'll subtype it and look for sequences in these H5 or these hemagglutinin genes. And nowadays, a lot of what they're doing in DVL is just sequencing the whole virus and looking at the sequence that way to determine if it has like those large chains of basic amino acid residues at the cleavage site that would make them highly pathogenic avian influenza. Now, for control, isn't as straightforward as the next disease we're talking about, but it's really multifaceted. And the primary form of control for this disease has been biosecurity. Um, Also, education, surveillance, and stamping out are other things. And these aren't one versus the other. It's a multifaceted approach that requires all of these. Vaccination is a really interesting one, and it's not very straightforward. We're actually going to use this as kind of a segue later on to talk about how this HPAI is different from Newcastle disease. But we do not currently vaccinate any commercial poultry for HPAI in this country. What we do know is that USDA ARS did start testing 5 candidate H5N1 vaccines in last year, 2023. And this is in the interest of stockpiling these and employing them during emergencies, like we would do with FMD, for example, which you might have talked about this morning. But the bottom line is HPAI vaccination comes with many challenges. You have to match it to a specific strain or subtype. They can be really difficult to implement. Cost is always a concern. And then maybe the last straw for most people are the trade restrictions. So a lot of countries won't accept your product if you've been vaccinating for HPAI. So again, it's important to note that all 5 of the vaccines currently being evaluated, they have near 100% clinical protection, according to some preliminary reports in chickens, but they still rely on a 2-dose regimen. And if you tell any producer, but especially a poultry producer, that there is a good vaccine, but it has to be administered twice, it changes from outbreak to outbreak, and you can't export your product if you use it, there's going to be some work that has to be done there. So, but it is on the horizon. So now we've reached our first knowledge check. I've talked enough for now, and I believe Liz is going to take it away with the first question, which questions are shorter.
There it is. There is a question in the chat too, Grayson, if you want to address that while we're answering this one.
Yeah, the question was regarding for non-poultry or commercial birds, which type of swab collection is best? And if I understand correctly the question, that's going to be those oropharyngeal swabs, just because they're easy to collect and they're almost always diagnostic. They're cleaner than cloacal swabs, and the virus is shed at really high levels in the oropharyngeal tissue too. So that's going to be your basic, your go-to sample is going to be that oropharyngeal swab, which we'll actually get to practice taking on live birds in the in-person training.
Okay, we're going to give a couple more seconds and then we're going to share the results.
This question is meant to be a segue to our next disease.
And I don't see where I can share my results or end the poll. Oh, there it is. Just give me a question. All right, here we go. We're going to end the poll. And these are the results.
Nice job. So the question was, HPAI infections clinically indistinguishable from virulent Newcastle disease infections in poultry. And that is true. You cannot tell just based on clinical signs and necropsy findings alone which disease you're dealing with. You really do have to do that diagnostic testing we talked about. So, and remember that, you know, although they cause the same clinical presentation, it's an entirely different agent. So now I want to give you an overview of Newcastle disease, a really cool disease, and then we'll wrap this up by talking about comparing and contrasting avian influenza and Newcastle disease. For Newcastle, we'll talk about the history and virology, then the epidemiology, followed by clinical findings. Spoiler alert, they're very similar, if not the same, to HPAI, but I have more pictures. And then we'll finish with control, which is a huge difference, the way we control this disease versus HPAI. So I like to start with history. When I was a kid, I was like really into castles. So that's kind of a preview of where this is going. But Newcastle, there were actually simultaneous outbreaks in 1926 in Indonesia, but also at this town in England called Newcastle upon Tyne. And in that town, there is, you guessed it, a castle. And it was new in 1177 when it was truly a new castle. And the only thing left today is the castle keep, or the stronghold, if you will. And when I was like a kid and reading about castles, I found out that the keep is kind of where you go as a last ditch effort when you've been compromised and you go to protect whatever's left. And that's how I like to think about Newcastle disease. It's endemic throughout most of the world, but it's our job as foreign animal disease investigators to keep it out of the United States. So we are essentially in the castle keep of Newcastle. And believe it or not, commercial poultry actually started to kind of explode in the United States around the 20s, and in Delaware is actually where it's supposed to have started, commercial poultry production. And in dealing with these outbreaks of Newcastle disease and avian influenza, right when commercial poultry production starts to get ramped up, the USDA was on top of things, as we've always been, and they started publishing educational material and promoting biosecurity. And I just love how creative this poster is from the 1920s, published by the USDA. It says, "Don't sell the laying hen. All springs should be turned insects, weeds, garbage, waste into eggs for the nation." And that's just them saying they don't want you to move chickens around. It's kind of the same way we control these diseases nowadays, is through biosecurity. So this is a virion of Newcastle disease virus. This was actually taken, this electron photomicrograph, by Dr. Clavijo, who is our NBAF director. And this was in 2000. And there was a bird that was in quarantine trying to be imported into Canada. And Dr. Clavijo recognized clinical signs and diagnosed it as having Newcastle disease. The import was not allowed, and therefore Newcastle disease was kept out of Canada. So just some fun history on that one. Here's a graphical depiction of Newcastle disease. It's the viral genome encodes 6 proteins, not 8, it's 6 as compared to avian influenza. This F or fusion protein is the most important and one we'll talk about next. And then this H and N is similar to the hemagglutinin neuraminidase protein in avian influenza. The nomenclature of this is a little bit more intense. So we'll just start at the top with the family. This is not an orthomyxovirus, it's a paramyxovirus. And if you go down to genus, you'll notice some other famous genuses in the Paramyxoviridae, such as morbilliviruses, which causes measles in humans, but we know as veterinarians it causes rinderpest, which has been eradicated from the world. But Newcastle disease is an abulavirus. We could further sublocalize that to serotypes. So all Newcastle disease causing strains are APNV, avian paramyxovirus serotype 1, but there are other serotypes, but Newcastle disease is constrained to a single serotype. And then finally, we get to our pathotypes of APMV1. And this is where velogenic, mesogenic, and lentogenic comes into play. So we are most concerned with velogenic Newcastle disease. So if you basically just follow this graphic on the left in the gray boxes, you'll see that you'll finally get to where we're worried about. And this is the pathotype of Newcastle disease that has high virulence, up to 100% mortality, and severe disease. Now, these other pathotypes of Newcastle disease are where an important control strategy for Newcastle arises that I'll talk about later. But this is the nitty-gritty about the nomenclature for Newcastle disease. So here we are, we finally arrived at virulent Newcastle disease. I've abbreviated as VND, but it's also been known by many other names, including exotic Newcastle disease, ironically named END, avian pest, avian distemper, again, just because it's similar to some of those other morbilliviruses. We can even further sublocalize it to the neurotropic and viscerotropic virulent Newcastle disease. So this is a video of a chicken that has been infected. This is at Plum Island with the neurotropic version of virulent Newcastle disease. And there's no audio to this, but you see these chickens, you know, you look in the cage and you think they're dead, but you realize they're still breathing. They're just completely paralyzed because of how it affects their central nervous system. So it's pretty obvious when you have this in your hands, although less common, I would say, but this is one place where you might be like, okay, this may be Newcastle disease if you see this and you see organ hemorrhages, for instance, because HPAI can present with neurological signs, but not necessarily as dramatic as this. The one we're more likely to see is this viscerotrophic form. And you'll see when I go over the gross lesions that this just produces hemorrhage everywhere, but especially in the lymphoid dependent tissues. And in birds, those happen to be the cecal tonsils in the digestive tract, as well as Peyer's patches and other places where there are aggregates of lymphoid tissue in the bird's body. So here's the overview of the regulatory part of virulent Newcastle disease, and WOA defines it as reportable Newcastle disease as an infection of poultry caused by Newcastle disease virus, which again is APMV1, and it has to meet one of the following criteria for virulence. So similar to HPAI, the gold standard for Newcastle is actually intracerebral pathogenicity index in day-old chicks. That is the gold standard, but it's not our go-to nowadays. Our go-to nowadays is PCR and sequencing of those protein gene sequences. So it has to have certain multiple basic amino acids in the F2 protein and a phenylalanine at 117 of F1 protein. What does that mean? That is an absolute mouthful. So let's kind of break this down. This is my course in basic virology for Newcastle disease, and this is very similar to HPAI that I talked about earlier. It's just different genes and different proteins. But first of all, the immunogenic proteins of Newcastle are the F and the HN proteins. This is how we do serological diagnostics for it. It's based on these proteins. But entry of Newcastle disease virus into host cells requires cleavage of this fusion or F protein. And in lowly virulent Newcastle disease, extracellular cleavage is only by respiratory and intestinal proteases. So it's a localized infection. This is probably sounding familiar. However, in virulent Newcastle disease, this intracellular cleavage is by ubiquitous proteases in many tissues. And it's because of these multiple basic amino acid residues at the cleavage site of this fusion protein. So that's it. Basic virology, basic amino acids at the cleavage site of the fusion protein that leads to systemic viral replication. We target this site for our diagnostic assay, which again is an RT-PCR that targets the F cleavage site, and that's how we get a diagnosis of virulent Newcastle disease by looking for these basic amino acid sequences. General epidemiology for Newcastle, this is a globally distributed disease. It has a wide host range, but only in birds. And it's been documented over 236 species of avian-like species. Has a 2 to 15 day incubation time, average of 5 to 6 days. I believe this is similar to FMD. And transmission is by oropharyngeal secretions and fecal material. So now we'll start with my favorite part, which are the gross lesions. Again, this is a clinically indistinguishable disease from avian influenza, but why not just dive into some other hemorrhage lesions? So this may present with severe lethargy, comb hemorrhages, and necrosis. Older birds, you may see different signs, or more mature birds, I should say. And one of them that can kind of clue you in is this periorbital edema. And again, there's, I'll show you a little trick on how to actually like look at this eyelid during a necropsy. And there's an aggregation of lymphoid tissue here that you can actually see hemorrhages in the eyelid if you look for it. So this is one presentation in older birds. And again, just hemorrhages everywhere. We have hemorrhages in the proventriculus, hemorrhages in the thymus and necrosis. We have hemorrhage in the cecal tonsils, which we saw in the beginning, and also hemorrhages in the larynx and trachea. Younger birds still have their yolk sacs. You may see hemorrhage and necrosis there. So on the left, these are just really necrotic yolk sacs with multifocal hemorrhages. And again, a different picture of hemorrhage in the larynx and trachea. So your main rule out here is going to be infectious laryngotracheitis if you're a poultry person. And these are some of my favorite images of, again, you guessed it, picking up on a theme here, more hemorrhage. You'll get to hopefully see this in-person training, but hemorrhage at this junction of the gizzard and proventriculus. Also, I showed this in the beginning, but this is hemorrhage in the GI tract at the cecal tonsils and the pyrus patches. And then these are the lungs with multifocal hemorrhages throughout them. So one of the things that is unique that I want to point out about Newcastle disease is that it's constrained entirely to one serotype. And within that serotype, there are a lot of different genotypes. But what that means is that all Newcastle disease virus strains, antibodies made to any strain of Newcastle disease can neutralize any other strain of Newcastle disease. And we can further divide these into different genotypes. But if you're thinking what I'm thinking, you're probably thinking, that must mean that this would be a great candidate to produce a vaccine for, and you would be correct. So here are a lot of different genotypes for APMV1, but remember they're all one serotype. So we have pigeon, cormorant, classical vaccine strains, and these were all distinct from one of the latest outbreaks we have to reference with a lot of genetic information about, which was the California virulent Newcastle disease virus. But again, all one serotype, just a lot of different genotypes. One of the things that's very interesting is the pigeon, and this pigeon paramyxovirus 1. It is reportable if this TPMV1 strain is isolated in chickens. And we actually had a really good discussion at our last in-person FAD training where the case was one of our people at the port at JFK, they received pigeons all the time and trying to be imported into the country. And if they isolated or they tested positive for Newcastle disease, they would actually have to do the gold standard testing at NVSL Ames to absolutely confirm that it was a virulent Newcastle disease virus. So this is kind of an interesting fact that pigeons can be a reservoir, and if it's detected in poultry, it's reportable. So does anybody know if we produce pigeons commercially in this country? Anybody ever heard of squab production? Yeah, so I had never really heard of squab. You know, I come from the southeastern United States where it's all chickens and turkeys. But on a trip to California, I got to get exposed to squab production. And here's me with a pigeon, and also got to go to a squab processing plant. So, and racing birds is the other thing I was going to say. You're reading my mind. Yeah, a lot of people race pigeons, and in Europe, this has been a really, really big problem and almost banned in some cases due to Newcastle disease and the risk of exposure to commercial poultry flocks. Anyway, kind of cool. Newcastle disease is a pigeon thing, and sometimes that overlaps with commercial poultry production. Which leads us to our next knowledge check. I've probably already given the answer away for this, but just to make sure you're still awake after lunch on a Monday.
Okay, I'm going to go ahead and end the poll and we'll share our results.
Excellent. All of you are paying attention. So again, the fact that all Newcastle strains belong to one serotype, that is APMV1, that makes vaccination way more practical for this disease compared to HPAI. So the way we control this disease is by live and inactivated vaccines that have been used in commercial poultry for a long time. There are many different options with different requirements and different shelf life and stabilities. Some are more common in the US, some are more common in other parts of the world. But these are all different pathotypes of virulent Newcastle disease. But never the velogenic pathotype is what it boils down to. So we can actually use this as a tool to give them protective immunity against Newcastle disease infection. This is in commercial poultry, right? And we know that commercial poultry is pretty structured, backyard poultry is the wild, wild west. Imported poultry, nobody knows. So it's way more likely that they wouldn't be vaccinated for Newcastle disease, which contributes to the outbreaks of Newcastle disease and backyard flocks, which I'll talk about next. There's no treatment for this disease, so prevention with this vaccine is the best option we have. So there's just a brief timeline of outbreaks of virulent Newcastle disease in poultry in the US. There was an outbreak in the 70s in California, a multi-state outbreak in 2000, and another outbreak in California that was relatively recently. Some of you may remember it. I know I do. And we have probably the most data on this 2000 outbreak. This occurred in California, Nevada, Arizona, and Texas. It included backyard and commercial birds. This is what made it different from the 2018 outbreak, which was entirely constrained to backyard birds. But this resulted in the culling of 3.16 million birds and an economic cost of $120 million, not adjusted for inflation. So this is a really big deal, especially if it spills over from a backyard poultry flock into commercial poultry flock. And while there are vaccines in commercial poultry flocks, we know that sometimes they may be more susceptible due to environmental conditions or stress, and that may be what happened in that case. This is just a quick note on how the clinical presentation can vary from outbreak to outbreak and strain to strain. So again, we always, almost always see with virulent Newcastle disease hemorrhages in those lymphoid-dependent tissues. But this is one case in the 2000 outbreak in California where they developed a field case definition, and it included a bird that had 2 or more of any 5 birds that had any of the following gross lesions. They were presumptively identified as having virulent Newcastle disease: hemorrhagic tracheitis, hemorrhage at the junction of the proventriculus, cecal tonsils, and then a diphtheritic lesion. And this is one of the ones that is a little bit more unique or specific to virulent Newcastle disease, just in case you ever see it. So the latest outbreak of velogenic Newcastle disease in the US occurred in 2018. And you can see that it kind of started with a couple of backyard farms in Southern California, and 2 years later it had spread to multiple farms, even outside of this quarantine zone. And it was declared over in 2020. But they really sent a lot of people out to manage this from 2018 to 2020. And exhibition poultry played a big part of this. If you ever hear that term, that usually could encompass certain illegal activities like fighting chickens and things like that. And I believe they were found a lot of spread was attributed to those activities as well. So we'll wrap it up with our last knowledge check. And this is something that's relevant to everyone who will be coming out to the in-person training.
Okay, can you see that poll?
Not yet. I can't. Maybe somebody else can.
All right, let me try that again. I don't know why it's not coming up. Hold on. It's not letting me launch.
Well, I know
That
the question.
is very strange. Yeah, and I know the question too. Yeah, I don't know why it's grayed out. Oh, wait, here we go. It just is slow. Very slow today.
There it is. What is the only zoonotic agent we work with during FAD training? going for 100%.
All right. Couple more seconds. Okay, I'm going to go ahead and end the poll. And here's the results.
Majority, 91% of folks got it right, and that is virulent Newcastle disease. So neither African swine fever nor classical swine fever are zoonotic, but virulent Newcastle disease is. However, in humans, it causes a self-limiting conjunctivitis, otherwise known as pink eye. So you'll see when we do this portion of the in-person training, we just wear some additional PPE, and that usually includes a face shield in addition to the goggles that we're already wearing. So it is true that when humans are exposed to high viral loads, especially like in their eyes, the virus can replicate in some of our tissues. So, but this is almost always self-limiting. It's just not something you want to have. And there have been relatively few cases of this happening. When I was reading about the zoonotic potential of Newcastle disease, I came across something interesting that kind of like threw me for a loop, and it said, intravenous injection of Newcastle disease produces more severe disease than like mucosal exposure. And I thought, who in the world would intravenously inject Newcastle disease? What human would do that? But that led me down another rabbit hole, and that is that Newcastle disease is actually being developed as anti-cancer therapy. And you can see as of late, it's even been looked to be a vector to express the COVID spike protein to result in immunity. So it's kind of just a fun fact about Newcastle disease, because we can mount a response to it and it can replicate in our tissues. It can be a good relevant application of other technologies and vaccines and such. So to finish it up, this is mostly for your reference. You will get all of these slides, but I just want to kind of wrap it up by talking about the similarities and differences between highly pathogenic avian influenza and virulent Newcastle disease. They're caused by different agents. So again, HPAI is a type A influenza virus. It's an orthomyxovirus, and it's further characterized by those H&N subtypes. Virulent Newcastle disease is just avian paramyxovirus, one serotype. Clinical presentation, again, remember these are clinically indistinguishable from one another, but here are the signs. Respiratory, you see coughing, snicking, some open mouth breathing and nasal discharge. Digestive, green watery diarrhea, and neurologic with depression, ataxia, and torticollis. And I think my footnote got cut off there, but ataxia is kind of more of a Newcastle disease thing. And of course, the most important one that isn't listed here is those hemorrhages and those different tissues throughout the body. And sudden death is always a good clinical presentation of these two diseases. So for your differentials, they're separate here, but just remember that anytime one's on your list, another is also on the list, and they'll likely test for both. Other differentials in poultry include infectious laryngotracheitis and some acute bacterial diseases, such as fowl cholera caused by Pasteurella multocida, can mimic the presentation of these two diseases just because it has such an explosive high mortality rate. Transmission is the same for both of them. It's fecal oral via aerosols, direct contact, or fomites. Diagnostics are different only slightly, and that with HPAI, we're looking for the H&N genes, and you can do sequencing or an intravenous pathogenicity index. Whereas for Newcastle, it's an RT-PCR for the matrix and fusion genes, that F gene I talked about, and sequencing or an intracerebral pathogenicity index. All of these samples go to Ames, Iowa, to the Diagnostic Virology Lab. And The zoonotic potential, we kind of talked about this with HPAI and pandemic flu and the potential for avian-like influenza strains to evolve into pandemic flu. So it can cause a mild self-limiting disease in humans all the way to a fatal pandemic-like disease. So this is a real concern for everyone. And I like to remind folks that before COVID, everyone was almost certain that the next global pandemic would be another influenza pandemic. So everyone was kind of caught off guard, but that doesn't take it off the table for the future. Something to be aware of. Newcastle disease, however, just causes a self-limiting conjunctivitis in humans, unless you inject it intravenously, in which case it might cure cancer. Who knows? But handy resources, they'll be in the slides you get. And your only homework before the in-person training is to review this work instruction, because this is what we'll really focus on in the in-person training. We'll practice taking these samples and how to submit them and CBL. So with that, I have about 8 minutes left, and I'd be happy to take any questions or talk about anything, or else we can end today early and we'll see you again tomorrow.